Hey everyone, good morning. Nice to see you again. My friend and colleague, Jeffres, is here today, and he's going to be talking about stable isotopes, floodplains, and his work in the Amazon, as well as here in California. And Carson is a research scientist at the Center for Earthquake Sciences, where he's worked for, I guess, since its inception, nearly 20 years.
Since 1999, as an undergrad.
That's right, he's gone all the way through. So he's got some great stuff for you. I want you to take note of the fact that I dropped a couple of the readings in the module for next week. There's 2 readings on rockfish, which we just published yesterday in Science, talking about the longevity of them. And I'm going to discuss rockfish next week. We're going to kind of go back and finish up our taxonomy and then go into fisheries biology for a few weeks, and that kind of is our entree to age class structure, longevity, and management of fisheries populations. So please check that out. Next week we'll have a quiz on Friday, and what else do I have to tell you? Topics. Got to pick topics for your blogs, and so really it's due today. But if you need to talk to me or you're unsure, you can continue to submit for next week. But the sooner you get it set up, the sooner you and your group can move forward on it. It's really, we just have a few more weeks left. So we're kind of in the final wrap here. Yeah.
Was that the 11:59 p.m.?
Yeah, but we're not going to hold it. You know, I'm not going to be sitting there waiting. So, so if you get it in the next, today or the next couple of days, or early next week, that would be good. It really behooves you to figure it out. Okay. All right. That's it. Carson, take it away.
Can everybody hear me all right? All right. Perfect. Thanks for having me, John. This is wonderful. I took this class as an undergrad in 1998 from Peter Moyle, and it really changed my life in a lot of ways. When I first got to Davis, I was an aeronautical and mechanical engineer for the first year, and then I realized that my true passion lies in fish. And so I took this a long time ago. Now I was a lot younger then and probably had more hair. But so it's fun to be here again. And you guys are talking about rockfish here soon. I imagine that the age structure is done with otoliths. And so one of the things that I've been really into lately is looking at biomarkers and how different parts of a fish's life are incorporated into various parts of the fish, whether it be otoliths, the ear bones of the fish, where time is recorded, or what I'll be talking about today is eye lenses of the fish, the lens of the eye where their diet is recorded. And so I'm super excited that you guys are moving this on. And I'm also going to say that I consider myself a proper fish squeezer. You know, fish are where my passion lies. But as I've gone through my career, I've realized that you have to study so many other parts of the system to understand how they affect fish. And so what I'll be showing today is some work that we've been doing on floodplains, both in California, as well as some work that I've been doing in the Amazon prior to COVID. Been harder to do such things after COVID, but so I'll show you some of those, and then I'm going to end it with this kind of some pictures from a 3-week expedition, the fish sampling expedition while I was down there, and just thinking about the fish diversity, the taxonomy is kind of mind-boggling. It's worth just showing you guys some of that information. So with that, we'll go forward with floodplains, biomarkers, and change. And so first, let's talk about floodplains. So floodplains are the section of land that's next to a river that inundates seasonally, particularly during the wet years or storm events or wet seasons. I'm sorry. I move a lot, so this might be challenging. You know, either rain events or the wet season of the year, floodplains will inundate, the water spreads out. And it's a really important time for lots of fish, lots of bugs, birds, bats, trees, use these habitats as they flood up every year. But it doesn't work well with people in that this floodplain is also very flat. It's a very fertile place. And it, on the surface during the dry times, it looks like a really great place to build cities and towns and agricultural lands. But the reality is that you end up in situations like this. You know, there's a painting from, I think, Marysville in 1864, in that lots of the cities in the Sacramento Valley, Sacramento-San Joaquin Valley, were placed next to rivers because they were the hub of transportation. Goods and people moved up and down through the rivers on boats. They were where rivers came together. You know, Sacramento is where the American and the Sacramento River come together. And so they were a logical place to build cities, but it turns out it wasn't very sustainable. And because of that, we really changed the system a lot. And that we built dams in the upstream parts of the rivers to collect that water before it came down into the Central Valley. And this happens all over the world. And then we also built levees next to the rivers to prevent that water from going laterally into the floodplain. And that really a levee next to the river is just basically a dam that runs sideways in a lot of ways. And it disconnects the river in the same ways that a dam does longitudinally, just laterally. And so because of that, we've lost most of the floodplains in California, really most developed parts of the world, we've lost floodplains. If you look globally, is that we've lost about 90% of that habitat, in the developed world. But to think about what happens when we disconnect the river from the floodplain or the river from upstream sites or because of dams, is we go into some of the kind of these conceptual models of how energy moves longitudinally and laterally through rivers. And so I'm going to just talk about these 4 concepts real quick. This is, we spent an entire quarter talking about the river continuum concept. Like we can study this thing for an entire quarter. And so we're doing this in a couple of minutes now. So for further reading, you can go down each of these paths. But the river continuum concept is an idea that energy flows differently longitudinally, depending on where you're at in a river. And with that, the fish community, the invertebrate assemblage, all those things change longitudinally from the headwaters really up high, where you have really narrow streams, you have leaf litter that falls in. The animals that are there are adapted to that. The insects are the ones that tear apart leaves, and then the fish are generally cold water species that eat those insects. And as you go downstream, your carbon sources change. As all of a sudden your rivers get really wide, your energy isn't just falling in, but it's actually being incorporated, or it's being produced inside of the river. There's phytoplankton that's growing. There's zooplankton that's eating the phytoplankton. There are planktivorous fish. And so you can see that there's predictable changes in rivers longitudinally, is that you have generally like the trouts up high, you have kind of these intermediate fish in the middle, then you go to these kind of big river system fish as you get downstream. So the riverine productivity model is another idea that kind of thinks about where in a river your energy comes from. Oftentimes it's, it's those edge habitats. It's where you are able to have sunshine that is able to penetrate into the substrate to where algae can grow. And as you move throughout a watershed, as those places of productivity change, similar to the river continuum concept, but it's more looking at specific points of productivity, not necessarily the carbon sources in theirs. And the river wave concept. And this one I actually have, to be honest, I have problems with all these, and I think it's all good concepts. They're a generalization with lots of problems, but it's a good place to get your head in a spot to start questioning those processes that are happening. And so with the river wave concept is the idea that when you have a flow event, it moves through the system differently. Up high in a watershed, it's generally steeper. The water spends less time in that area, and there's more energy. There's less productivity happening in situ there in the river because the water and energy is moving so quickly through. And as you get down farther in a watershed and that water spreads out, is the amount of energy that's in that water and in that system is reduced. There's more opportunity for the water to interact with vegetation and break down vegetation. There's more opportunity for the sediment to settle out and have algae growing. And so you end up with this different concept about in different spaces in place and time, the amount of energy, stream energy, as well as food energy in that system. And so the flood pulse concept, and this is really, I've spent a career at this point on this concept, is that this is really what got me inspired to start thinking about the work that I'm doing on floodplains was the flood pulse concept. And it's an idea that when water floods up, when the river stage comes up, the water spreads out, and with it goes energy. And energy is going in 2 ways. Energy is going from the river into the floodplain, and that oftentimes that river water during a flood event has lots of nutrients in it. It has nitrogen and phosphorus, and there's a lot of things that make life happen. And that when that spreads out, there's that life that goes out into the river or out into the floodplain. It goes into the trees, it goes into the grasses, it goes into all the vegetation. And there's a lot of productivity that happens. And when all those nutrients are out there, you have a lot of photosynthesis that happens in the floodplain. but then all those nutrients get used up. But what happens is that now that that water has time to spend out on the floodplain, is it gets cooked. It's like steeping tea, in that when you take a tea bag and you put it in water and all of a sudden you see all those dissolved organic carbons come out and your water changes color, that's the same thing that happens on a floodplain. Is that when water spreads out and it interacts with all of that vegetation, whether it be leaves that have died and dropped, which are a lot like tea leaves, it turns out, grasses that have died throughout the summertime, they've grown in the spring and senesced during the summer, is they release their organic carbons. And they're steeping the tea. And it turns out that there's so many organisms that can use those organic carbons that are released into that water. There's microbes, there's zooplankton, there's bacteria, there's single cell critters running around out there that are eating all these things. And it ends up being a super productive environment. And what's cool is that there's so many fishes that have learned to take advantage of this environment. Is that over millennia, fish have evolved to move out onto the floodplains to reproduce. They can lay eggs out there. They can go out during their migration to utilize these food resources that are growing out there. And it ends up being this hyper-productive environment where just magic happens. I mean, in lots of ways, it's kind of amazing when you go out and sample the zooplankton on the Yolo bypass. Who's familiar with the Yolo bypass? Everybody, you drive over on the way to Sacramento. Who has seen the Yolo bypass full of water? Some people. The Yolo bypass fills up with water. It's not just an agricultural field. When in 2019 was the last time that it was full of water, there are millions of salmon that are moving through there. It's amazing. You drive to Sacramento and there's salmon swimming underneath your feet, underneath your wheels of your car. And they're swimming out there because there's so much food. There's about 300 times as much zooplankton in the Yolo bypass than there is in the Sacramento River. And so when that water spreads out and all those things happen, it creates an environment for fish that are spawning out there. Have you guys talked about splittail yet? Yeah, so the Sacramento splittail, it spawns out in these floodplains, and when the young hatch, they're hatching out into an environment that is full of food. It's super productive. And there's a reason why they evolved that life history, is because they're taking advantage of these resource pulses that are happening during the flood pulse concept. And like all good concepts, this one's wrong. The flood pulse concept really focuses on primary production. It focuses on that algae that's growing out there and the phytoplankton. But there's also a huge heterotrophic pathway as well. So all the microbes that are using that dissolved carbon, all the bacteria, all those single cell animals that are using these other pathways are also really important out there as well. So thinking about the flood pulse concept, it really was started in the Amazon and the Orinoco. And so if you think about it, the Amazon is the largest river in the world. At any point in time, 1/5 of the world's fresh water is tied up in the Amazon River. 1/5. It is absolutely bonkers how much water is in there. It's hundreds of miles wide at flood. It's from the Sierras to the coast wide every day. So if you drove from San Francisco up to Tahoe, it's about that wide at flood. It goes up about 20 to 30 meters as the stage difference annually. 20, 30 meters, that's 60 feet. That's 6 stories of difference in stage on an annual basis in places. It's truly amazing. And so when Jean and Bailey, who were kind of the first people who described this concept, they were doing work in Amazon, they realized that when that flood stage happens, is all the fish left the river and would go out into the forest. And there's all this productivity happening. And let me see here. And so what happens is that water goes up, spreads out, the fish happen, everything is just wonderful. And this is kind of a good diagram that came from that original paper, is seeing that and just imagining where conditions are at when the water is all stuck within a single channel in a river versus when it spreads out, and the difference over space and time and how animals and energy move laterally, not just longitudinally through the system. But here we are. We are humans. We're very good at disrupting natural processes. We see that on a global scale. We have fundamentally altered the Earth in more ways than we can possibly imagine. And one of them is disconnecting, and we're just talking about one of them today, is disconnecting the floodplain from the main channel river. In California, it's very obvious what we've done, and that when you drive over Sacramento, when we drive to Sacramento, you drive over the river, and it's like, what, 4 or 5 seconds of driving over the Sacramento River, the largest river in California. And it's kind of crazy to think that we've taken this giant watershed and confined it all into one channel, just so we can build houses and cities. I mean, we all benefit from these things. It's not like it's terrible, but there's ways that we need to think about how we can take these disruptions in natural processes and kind of think about the ecosystem function that happens by having them back in place and thinking about the restoration side of it. But it's not just in California. This is the river that I worked on in Brazil. This is the Xingu River. It's a tributary to the lower Amazon. And what you can see here is this is the Xingu in May of 2000 on the left. And there's the Xingu in July of 2017 on the right. And you can see that Pimentel Dam was, I'm going to walk over here and I'll point this out. Pimentel Dam was constructed here, and it's not very tall. It's only about probably 40 or 50 feet tall, which from a dam in California, we think of 700 foot, Lake Shasta Dam is 700 feet tall. And here we have like a 50-foot dam that is gone, and it didn't make a big reservoir. It made a little bit of a reservoir. You see here, there is a reservoir. But what they do is they can divert the water through here into an artificial lake and then dump it down into Belo Monte. And so Belo Monte, you see up there, is the world's 4th largest hydroelectric facility. It is absolutely gigantic. And so there's 3 gorges. There's Itapu, which is on the Paraná River. And then there's, I can't remember the third one, but it's in China. And then Belo Monte is the 4th largest hydroelectric facility in the world. And so closure of this dam happened in 2016. And this 75-kilometer reach from here, from Pimentel Dam, they call this Volta Grande, the Big Bend, goes through, it's mostly bedrock. And it's lost its floodplains. Is that now they divert the water through here to generate electricity. And I'll talk a little bit about this in a little bit, but there are a bunch of indigenous tribes who lived in here, and the fish in this section of the river are their livelihood. That's where most of their protein comes from. And so there's been a lot of work trying to figure out, quantifying what are the effects of the fish assemblage, changes of the fish assemblage. How have people lost their livelihood, and what do you do about it? What do we do when people all of a sudden lose their economic base, their food, and how do you manage that from a system? And can you actually regulate this dam to make it more beneficial to the fish and the people in the lower watershed there? So how do we quantify the change? This is one of the things that when you ask somebody to give up money for something, so by diverting water around the power plant, you're asking somebody to give up money. You're asking the company to not make as much money as they were doing. And so you have to be able to quantify how much bang for your buck you get. So let's say we let 5% of the water pass. Do we get more fish out of it? How many more fish do you get for that use? And so fortunately, we have been developing some tools to actually get at this. So this is Miranda Beltelcock up here on the top. If anybody has extra time on their hands, they have 12 minutes, I think is how long this is. She was on Science Friday in January talking about this work. And Miranda was a student over at Watershed Center. And basically what we're doing is we're trying to quantify the changes of floodplains in California. And these are tools that we're ultimately applying to some of the Amazon work as well. And the idea is that in different points of the landscape, at different points in time, you have different isotopic signatures. And so what's cool is that when you see up here that there's this, wetlands and floodplains, this is delta 34S. Just think of this as sulfur. It's a sulfur isotope that has a unique signature. That's all you need to know for right now. Wetlands and floodplains have a value. The river has a value. Hatcheries have a value. And the ocean has a value. If we know the different values of different locations, we can start looking at the landscape and saying, okay, this is water samples from the Sacramento. And you can see that YOLO has this different signature than the river does. And so we were curious about whether we could start thinking about how we can quantify those differences in fish. Then first we started looking at the stomach contents of different fish. And so when we look at the carbon isotopes and the sulfur isotopes, we see that the yellow dots are one group and the blue X's are another group. And so okay, so we have something that actually we can quantify the difference in the stomach contents of the fish. But stomach contents are a short-lived thing. in there for a day, maybe. A fish eats something and it poops it out and then it moves or stays or does something. But if you look at a fish's stomach contents, that's only what was there on that day, which is important because you need to know more than that because we need to see whether they've been there for a while. And so Miranda's work was looking at these different tissues of the fish. So here we have the stomach contents in the blue line, and then muscle. So if you look at the muscle of a fish, it equilibrates to the stomach contents. So after about 30 days, the muscle tissue looks the same. So muscle tissue in a fish changes about every 30 days. So you are what you eat, right? You've heard that saying? It's definitely true in fish. If a fish is eating something, it looks like that isotopically. If it changes food, it also changes, which is really useful unless you want to know what it did more than 30 days ago. And so we started looking at the lens of the eye of the fish. So you can see that the lens of the eye also starts looking at like what the food resources are. And what's interesting about the lens of the eye is that as the eye lens grows, it grows out, and as the fish gets bigger, it puts on more and more tissue. And so on the outer end, this is the lens of the eye of the fish right here. This is what it looks like when you cut it in half. On the outer edge of this are epithelial cells, like the edge cells, and they're growing and growing and growing. And then what happens is kind of awesome. It's not kind of awesome, it is awesome, is that all of a sudden it gets to a certain point and then it changes structure and crystallizes. And what's cool when that happens is that when it becomes that crystalline structure, is then it no longer changes its isotopic value over time. So it has then become what it was at that time when that eye lens formed, when that eye lens layer formed. And so it is no longer what it's eating now, it's what it ate while it was growing at that point in time. And so what's beautiful about it is there's a ton of cysteine and methionine in these eye lenses. So there's a ton of sulfur. You see these sulfur groups here? These sulfur groups are incorporated into the layers in a big enough amount that we can take this eye lens, we can peel it like an onion. We can peel each of those layers and look at what that salmon was eating. It's basically a diet journal. And so that the fish are recording their diets over time, and it kind of looks something like this. Is that, let's just say that the floodplain is yellow, the river is blue. These are delta, they're just the relative values. You don't need to know what's happening, but we take the eye lens, this is actually what it looks like. We peel it, we open it up, we put it in little tin capsules, we send it to the isotope facility, and they give us data back. And then we'll take each of these layers and see whether they've been, this is mom, right? So they're an egg. You are at the beginning what your mother gave you. So this is mom, this is river, this is floodplain, this is ocean. And so if you look about it through the lifetime of a fish, as an adult salmon, you can go look at each of those layers and see what it was eating throughout its lifetime, which is pretty cool. If you read its diet journal over time, you can start to look at different patterns throughout a bunch of fish. So you can start to look at the population, how they're using these habitats. So what we found is that a lot of the fish are using floodplains. So these are fish that these were adult fish. So these were fish that were born in the river, high up in the river. You guys have gone through salmonids? All right. So these fish are born up in the river. They migrate out during their first year of life. They go to the ocean. They come back. These are all 3-year-olds. So they come back 2 years later. And so when we collect these fish, these are winter run. These are the wild winter runs. So these are the endangered species. There's hundreds of them left. There's not many. They're not doing very well. But we can see that the majority of them that come back are using floodplains during their outmigration, which is pretty cool. We can start to look and quantify the use. If you look at the hatchery fish of the winter run, none of them use floodplains. They release them late in the year when there isn't floodplain opportunity available. And so we can start to look at this and give this information to people who are making decisions about restoring floodplains or releasing water. And we can tell them how much benefit they get on the landscape when this happens. A fun fact here. So these are the adults that went out to the ocean. Granted, we have a bunch of fall run data. Almost none of the fall run juveniles have floodplains in their eye lenses. So there's a disproportionate recruitment into adulthood. So even though a few of them are using floodplains when they're juveniles, when they come back from the ocean, the majority of them have used floodplains during their juvenile life stage. So it's a nursery habitat. This idea that there are habitats on the landscape that recruit disproportionately are out there. And so these floodplains are doing it. It's so cool. And so we're working with folks now to say, hey, this is really important. If you're going to spend your money, if you want to save, some of these endangered species, that this is one of the tools that you can put in your toolbox to figure out how that actually works. So how do I translate that to Brazil and the Amazon? I'll get there, I promise. But I do have to throw a shout out here to the Fulbright program. So I had a Fulbright fellowship in 2019 where I packed up my wife and my daughter, who's sitting right here, who has a day off of school. And so this is what happens. And we moved to the city of Belém, which is at the mouth of the Amazon in Brazil. And then I was talking with John. He's like, well, how did you get this? I just cold called a professor in Brazil. I sent him an email. I was like, I have this idea. Like, what do you think? It's like, great, I'll give you a lab space. You go out on the expedition. He's like, I'll cover all the expedition costs and go forth. And it was, it's truly one of those like leaps of faith where you send random emails to folks and you end up getting it. And then, I had that idea, and then I reached out to Fulbright and I put together a proposal. It's like, I really want to do this. It'd be kind of fun. And I got it. And it's, you have these random points in life where you make decisions and you mail in your box tops. And every once in a while you win a prize, but you don't win any prizes unless you mail in your box tops. So I just want to throw a shout out for Fulbright here because there's a ton of undergrad opportunities. There's a ton of graduate opportunities to work and travel all over the world. There's Fulbrights in most countries in the world, actually. I was with Fulbright Brazil, and it was really amazing. They're super supportive. If you are interested in any sort of international work, even Canada counts as international work, it turns out. Like there's a whole Fulbright Canada group, which kind of blows my mind because I just think of it as the 51st state in so many ways. But there's a lot of opportunities. I just wanted to make sure that put a plug in there for Fulbright because it's really cool. And it was honestly the hardest thing I've done in my life and also the best thing I've done in my life. And so I would recommend just kind of, stepping out of your comfort zone every once in a while and thinking about doing things like this because they can be really rewarding. All right, so what was my project in Brazil? So I showed you guys this picture earlier, is that this is 2000 and pre-dam, just think of it as pre-dam and post-dam. And that my question is that can I use these fish, this is peacock bass, for Tucunaré, you'll see it also listed, that we would go out and collect. They're relatively stationary fish. They don't move a long ways throughout the river. They move laterally between the floodplain and the river as it floods up and spreads out. Can we go out and collect their eyeballs, which is a really weird thing to ask. I will show you some pictures of trying to convey this experiment to the indigenous folks in my bad Portuguese. And then can we look at those patterns in their eye lenses and look at changes in the patterns of the eye lenses to quantify the difference between here and here? Because what's interesting is that when you look into the eyes of a fish, you can see its story. And that because we collected these in 2019 and the dam was closed in 2017 or 16, is that we actually had like 5 years worth of data in those eye lenses. Because it goes back in time throughout the ontogeny or the life of the fish. You can see those patterns in the eye lenses going back in time. I'll present a little bit of data like this for this, but the rest of the data is currently in the queue. I'm bombarding them with data at this point. So, but this is kind of the general idea of what I want to do, is that if you look throughout from the beginning, when this fish is born to when we collected it, and these are fake numbers up here, so take this as you will, but you can look at each of those values across, and you know that the dam went in right here, is that those inner layers actually account for the time before the dam was in place. Accounts for the outer layers are for when the dam was, after it was built. So if we look at sections both above and below, and we go out, we collect fish in the field, we go back to the lab, we separate the layers, each of those layers, these fish had some of these fish had 45 layers of eye lenses, which is brutal. You sit there with a little tiny pair of forceps and you peel layer after layer after layer after layer for both eyes, because you have to do both eyes. You do that in the lab, you put them in the things, you take them to the mass spectrometer on campus, and then you run them for isotopes and look for the signatures that we talked about earlier. And the idea is this, that this is the conceptual model that I put together that is probably wrong, like all good conceptual models are wrong, is that you see a cycling of sulfur up and down. These are the flood times. The sulfur is lower in flood. This is a non-flood time. So dry season, wet season, dry season, wet season, dry season. Oh, but we lose our flood pulse below the dam. And so all of a sudden we lose those sulfur values below the dam. And so this is one of the things that we're really trying to see. And all my sulfur samples are actually in the queue because I honestly didn't know if it was going to work for any. And so I ran some other samples first I'm going to show you, but the idea is this. I want to show you that this is also, you saw Amber's beautiful visuals before of the river continuum concept, all the different concepts. This is my visual art. I use art loosely. But you can see that there's a dry season water and there's a flood season water. And this is what is currently happening, that during the dry season, the water is within the banks. During the wet season, it goes up high. This is the art of downstream areas below the dam, is that the river is lower in the dry season and it's also lower in the flood season. So you don't actually get as much interaction. But then what's crazy is that this is something we hadn't accounted for when we first did this, is that above the reservoir, even though it's not a very big reservoir, is that there was still a ton of trees that they cut down. They went to the forest, they cut down all the trees, they put a dam up, and then it backed up and made a lake. And what's crazy is that there's a huge change in how nitrogen moves through the system when you build a dam. And we hadn't accounted for this, and we thought about using our baseline. We talked about there's predictable values on the landscape. This is something we hadn't accounted for. And it turned out that it was pretty interesting. But to understand why it's interesting is this is a basic isotope primer. And so this is carbon, this is nitrogen. Notice there's no sulfur on here. Sulfur does not change as it moves up the food web, but it is more of an indicator of various food webs where it's at. So as you start with plants, you go to small fish, you go to bigger fish, bigger fish, bigger fish. There's a predictable pattern that you see here, is that nitrogen goes up about 2.5 values per trophic level. So a plant is here, a fish that's eating plants is about 2.5, 3 higher. The fish that are eating the fish that are eating the plants are another 2.5, 3 higher, and anyway, so on and so forth. So the idea is that if you go out and sample fish, and you've been sampling fish for a long time, like, okay, we caught this fish, it is a 3rd trophic level consumer. So it's eating the fish that's eating the fish that's eating the plants. It's at level 3. But this assumes that things are consistent over time. And that's not really the case, it turned out, and we didn't necessarily appreciate it. Part of it is because this is what a real food web looks like. So I showed you what an idealized food web of isotopes looks like, and this is what it looks like when you start modeling all the connections in a food web. It's a mess. There's food, everything eats everything else. You know, I think most fish are opportunivores, that they eat what they have the opportunity to eat. And the reality of it is it's quite messy. But that's still assuming that you have a stable baseline of information. So when we go through here and we collect all this data, this is 5,000 isotope samples from fish. That's a lot of fish. It's quite amazing to think about how many samples they're collecting on an annual basis. And we'll talk about the sampling program in a second. But this is a lot of data. And you see that from a carbon perspective, it doesn't really change much. Either downstream of the dam, and also the dam went in 2016. So this was the year that changed. You see a little bit of bump there. Upstream of the dam, carbon doesn't really change. But what's crazy is you look at nitrogen, it goes up like an entire trophic level. And what's interesting is these are the same fish, the same species that were sampled throughout this time period. And they went, all of a sudden you're like, let's just say you have a rainbow trout. I'm making up, this isn't in Brazil. But let's say the idea would be that you have a rainbow trout. It feeds right here. You put a dam in, all of a sudden it looks like a rainbow trout is eating a bass. You know, all of a sudden you change trophic positions across the entire community of thousands of fish samples. And all of a sudden everything changes. And it's super confusing because now, as I told you, I have gone back in time with my eye lenses, right? And that I thought I had this great idea. I was going to be super smart and famous, and I'm not, because everything changes in unpredictable ways. When you're thinking about using isotopes, it's assuming that you have constant systems that you can compare to each other over time. But I talked about the sulfur, and now I'm still waiting for my sulfur samples to get back because sulfur doesn't fractionate trophically, which is super important, and that we'll be looking at those actual baseline samples that are happening over time because it's more consistent. So when I got back, I got back right as COVID started, which is a really hard time to do lab work and submit samples to a lab that's closed. So this is our first batch of samples. So each one of these dots is an eye lens layer of various fish. There's only, I'm just gonna talk about this briefly because this is a limited set of data that we have and the rest of it's over there at the isotope facility on campus. But there's two things you do notice is that the dam went in about right here on these fish at about layer minus 25. And so what's interesting is that the purple line here and the red line, these two sites are the two sites in the reservoir upstream, is that their carbon values changed during that time. The green and the blue are the control sites, or the green site, which is upstream of the dam, which doesn't have any reservoirs, and the blue is the site below the dam. And again, you see those two separate differently between here as well, trophically from a nitrogen perspective. This is just a little taste of what I cannot wait for my sulfur data to come back because I think that's where, remember my conceptual model, things going up and down? That's what I want. But you can see that if you look back in time, there is something that's changing in the system. I don't know what it is. I'm waiting for more data. Hold tight. But what's interesting is that this fish program that I was part of is that they started before the dam was constructed in 2012. Is that they started going out twice a year. Each expedition is a month long. So they go out and they're on the river for 4 weeks sampling fish. It's quite impressive and it continues to today. And they do one in the flood season at high water and one at the low season, or the dry season at low water. They use gill nets and baited set lines. So we'd set out large batteries of gill nets. You guys talked about sampling methods? So gill nets, the fish are swimming in. What's cool is that they have 7 different sizes of net in that battery, from small little mesh holes to where small fish are getting in to big mesh to where you're collecting big fish. And then the set lines are on 12, so the hooks are about as big as my finger and they throw a whole fish on those hooks for fish. And then the other thing that's crazy that I actually didn't put in here is they collect via snorkeling. It's the most crazy thing I've ever seen. Is that they'll put masks, they actually don't snorkel, they just put a mask on and then dive and collect. And they have a 2-liter soda bottle with them with a slit cut into it. And it's tied around their waist. And they go through and collect fish off of the bentos and put them in, they push them and put them in that bottle. It's crazy. I caught 1 fish while I was out trying to do this. I was so proud of myself. And they're just bringing up 2-liter bottles full of fish every time. You know, these are people who have grown up doing this their entire life. So this is our fish sampling program. And then we'll go into a little more here. Since 2012, from 2012 to 2020, there's about 200,000 fish have been collected for this program. It is absolutely mind-boggling the amount of data that's available. Of that 200,000 fish, there are 449 species of fish. And it is crazy when they're down there and they can ID the genus and species of every one of those fish. Sometimes there's disagreement and there's a group discussion, but their taxonomy is mind-boggling. It's so impressive. And they go through and we get these big buckets of fish. So the fishermen go out, and I'll talk a little bit how we do it in a second. They go out, bring fish back, and then there's a discussion about every fish gets worked up. It's crazy. When you think about, there are more fish that we collect in this 150-kilometer reach than there are in North America, species-wise, by a long shot. So I went with this group of folks. There are fishermen, there are boat operators, there are cooks, there are people from the university. And we jumped in boats in the lower Xingu River. This is just a side channel. And we go forth. And I think before we talk about that, I think it's worth acknowledging the people who actually live along the river. There are huge groups of indigenous communities that still live here. There are people who live in their boats. You know, all of these boats are actually occupied by people. That is their house. You know, when we pull up we got ice every couple of days. There's a house, you know, every 40 kilometers that sells ice. And so we would have to get ice for our samples. And it's like, this is where these kids live. It's pretty amazing. And so we would run into these different groups of people throughout the trip. You know, the river is a corridor for goods. This is a ferry. So they put a car on the ferry. They navigate through this. You know, we're there at low water. I don't know how anybody could possibly navigate a boat with the amount of rocks in this river. But this is their livelihood, and we have functionally altered livelihood for large numbers of people, including the indigenous community. And one of the things that I said earlier is I never thought in my life that I'd be sitting in the middle of the Amazon in the middle of the night. This is like at midnight, explaining why I'm pulling the eyes of a fish out in my terrible Portuguese to a group of indigenous people. Like, of things that I thought in my life that would happen, that is not one of them. And so they came for a few days, actually they tagged along with us just to see what we were doing, because this is their indigenous land, and all of a sudden, folks from the university and these federal government employees, contractors are coming down and collecting fish. But what's interesting is that every night there was a different event. Like, all of a sudden, so we'd set the nets around 3 in the afternoon, and then we'd clean the first batch at 9 at night. But between 3 and 9 was kind of downtime, and various games would break out. They would set up a volleyball net in the water. We played soccer, football, in various communities that are they have these amazing fields that are lit up at night. It's a town of a couple hundred people, but they have their priorities, and volleyball and soccer or football are one of them. I also think it would be remiss to not talk about what's happening there right now. Is currently there's so much. This is a fire. This is where they were burning the jungle at night. We were on an island in the middle of the river, and this is the other side of it. And we could sit there and you could hear it burning all night long. And they're burning it for cattle and soybeans. And this is happening throughout the Amazon. And it's having a big impact on the water quality, on the lives of the people who live there. I have a friend who's actually on an expedition right now, and they're having a problem finding a place to camp because we sleep on the side of the river here in the trees, in the jungle. And all of a sudden, since they're cutting that down, they're actually having to take out and go to hotels. As where they're used to do these expeditions is that they would just stay on the river, and now they can't find a place to actually stay on the river because it's all being cut down and burned. So I promise I'll get happier. So here's the sampling. We use these long gill nets. They're about 300 feet long. We go out, and so these are fishermen who have grown up on the river. They fish their entire lives, and this is how they fish their entire lives. And then we had these baited long lines that would go out, as you can see on the right there. And then they bring back these fish samples. They bring back these big buckets of fish. It was fun. Like, we'd go out, and I was never allowed to clean the nets or clean the baited hooks because it's crazy. Like, I can't believe how they would process fish. One guy got bit by a piranha and almost lost his finger one night. Another guy had a spine go through his hand one night. It's truly crazy. But this is what we would do is, so we would get this would be our night sample. So these would come back around 10 or 11 at night. And we would process fish till about 3 or 4 in the morning every night. And this is what they have: a generator. They would set up the lights underneath the tarp. And you can see everybody's charging their cell phones. We have the computer set up. It's quasi roughing it. But what we would do is those fish would come, every fish gets worked up. So of those 200,000 fish, they've all been sexed. They've had a maturity index to see how mature they are, size, and then about 5,000 to 8,000 of them have had muscle tissue taken for isotope samples. It's pretty astounding, the process that goes through this. And this is what we did. And we'd also do another batch in the morning, but it's not nearly as exciting. And so now I think it's worth just talking about some of the samples. I got 6-7 minutes. These are kind of the iconic species. This is where we start talking about some of the fish. It's not just who's there. Who knows what this is? You can tell by the teeth. Everybody know what this is? Piranha. Yeah, they're everywhere. There's a whole bunch of species of them. The teeth are pretty amazing. You see videos of them and you're like, oh, that's a big crazy fish. And then you look at it and you're like, one of the guys almost lost his finger one night. There's the big catfish that are there. That was a 70-pound catfish that we caught on one of the set lines. It's truly crazy that the variety of fish you get. Who has an aquarium? Does anybody here have an aquarium at home? 1, 2, 3, 4, something like that. Does anybody know what a pleco is? Like the algae eaters in aquariums. Have you ever seen those? There are more of them than you could possibly imagine. There are probably 40 or 50 species of those in the Xingu River alone. In this 75, 150 kilometer reach, it is absolutely filled with this different variety of plecostomuses, what we call plecostomuses. This one was awesome. It was about that big, and its head was about that big around. And this was one of those moments where I should have put the picture of me kissing it. Is in my lab, there's a pride with kissing fish with fleshy papillose lips. And so we're in the middle, it's like midnight, and they bring this big thing, and I was like, oh, I have to take a picture of me kissing this. And they're like, there is no way you're going to kiss this fish. Yeah, I mean, how could I not kiss this fish? And there are several moments where you break the ice and just humble yourself that kind of endear you to the group. And that was one of those moments. So the Placosmises are really interesting. There's freshwater rays. So this portion of the Amazon is very old geologically. It's been around for a long time. It's relatively low. And so there's a lot of marine species that are in the freshwater that have evolved to take advantage of it. You've probably seen this in aquariums, the leopardi, is that this is an endangered species. So there are actually very few of these in the wild. And this is the only place in the world where they grow, where they live naturally, is in the Xingu. And they would catch them by hand. So I was telling you about, looking at catching fish, is they would actually catch them while snorkeling. And the fishermen would be going, they would see a ray, they'd take off their shirt and go down and grab the spine with their shirt and then pull them up. It was crazy, but it was so cool to be able to see all these different rays that you normally associate with being in a marine environment, and you are probably like 300 or 400 miles from the ocean. There's all sorts of crazy fish. There's, again, the piranha on the bottom right. This is something that's very similar to a paddlefish. Does everybody know what a paddlefish is? It's a filter feeding fish with a big bill on it. These fish have that same strategy, and they have really fine gill rakers that they actually collect zooplankton with as they filter the water. There's some of these big cats that move all over the place. It's a wolffish there that's found in the backwaters, is that they get really big, and do you think it's a predator? Oh, you look at body morphology, you can kind of pick out what most of these fish do for a living. And speaking of predators, the one on the left here is quite impressive. This is oftentimes called the vampire fish, and it has 2 slots in its head that its lower teeth go up into. So, and while fishing for other fish, you'll catch like piranha like this, and it'll come up with half of them gone, but you can see where the 2 holes ate the fish on its way out. It's quite impressive. There's freshwater drum. So this is a fish that's oftentimes associated with being in the marine environment that, again, has evolved to take advantage of it here. Bonus points for anybody who knows what this is. It's an electric eel. And what's crazy is that they are obligate air breathers because they evolved in this floodplain with a low oxygen environment, and it got caught in one of the gill nets. And so it drowned in the gill net because it couldn't go up and take a breath of air. It's not obviously what we want to do, but it was fascinating that you do get these bycatch of crazy things just by out there sampling. But it's not all just fish, is that there's a lot of other things. There's freshwater sponges and corals. This is a freshwater coral that's on the rocks. It's crazy. It's like a marine environment. But what's also weird is that there are hydrocyclids. You know, a freshwater caddisfly that you see in California is also in the Xingu River in the Amazon. There are beetle larvae. This is aquatic beetle. We would see something like this in California. But then there's also crabs. They're like crabs, like are out there like doing crabby things. Freshwater sponges are in there as well as these giant snails that are cruising around. It's like a snail. It's really impressive on the diversity of critters that are in the water in various forms. There are mayflies here. I don't know if anybody is into mayflies, but you think of mayflies as a cold water, clear water species. This water is no cooler than 85 degrees. It is hot, and yet it's full of mayflies. There's huge mayfly hatches at night. It's really unbelievable. But then there's also a whole group of terrestrial things that were just bonkers. You see praying mantises that look kind of like a praying mantis, but nothing else. This is a tent, and these 2 ladies were in it, and you can see the giant tarantula crawling on their tent. The giant river otter, it's about a 7-foot otter. I have no idea what that is. Caterpillars, don't eat that. I wouldn't eat that. There's crazy moths. But yeah, so I think that's more or less where I'm wrapping up because I think I'm about out of time. And this is just kind of your go to bed talk. I took a picture of my hammock set up in the jungle every night. And that it's just, again, it was a humbling experience. I'm an outdoor person, and this was a place where I've never felt more uncomfortable. I didn't even know how to set up a hammock when I got there. And I was that person where I had to learn and be out of my comfort zone. And I think that's it.
weekend. Thank you.
That was great fun.
Awesome.